how we, um, where you can find us and, what you need to get started with some of this research. Um, you know, from there, what I hope to have time for is really a conversation about, utilizing our samples, getting the most out of these precious samples. And, many of you, I'm sure, have already done human tissue research, but taking a step back and, thinking at the simplest level, how are some general ways we can approach this to get the most out of it? And I'll tie that in with some of the initiatives that we're approaching in the BSR. So ways we're increasing our collection value and bringing in advanced technologies to really help, guide this data collection and the analyses done on these tissues. So I'm clicking right, yeah. All right, so yeah, we'll start, not to downplay this, but boring, and we'll build up from there and get into the science. But we now have a physical space, right? It is exciting. We're located right off the yellow elevators on the 6th floor. So when you get off the elevators, we're putting up signage and whatnot. But we're in the 6,000 corridor. And really, where we're set up to work, we have this staff room, for our data management, for accruing clinical data to annotate our specimens, a really nice wet processing lab, and a really good sized storage facility for liquid nitrogen, storage, -80 storage, and of course FFP blocks, which I'll talk quite a bit about later. And then the satellite room off of our storage facility, that is where we're bringing in some of these advanced technologies. So we kind of in that room want to build, a spatial analysis center to an extent. And most importantly, I don't have any text or pictures for this, but we now have staff. And, that's very important to me. So we've recently brought on two full-time staff, Irina Stefankiv and Zoe Tomlinson, who are, really picking things up fast. So, we're really finishing off this build and getting going with our day-to-day. And also Jenny Twardowski, a PhD student in Stefano Mello's lab, is interning with us as she finishes up her PhD to learn more about working in the shared resources and in human tissue research. So all of them have been great and really seminal in getting this off the ground and getting to our day-to-day operations. And finally, not to cop out, but everything I talk about today, there's even more detail on each topic on our website. And to get there, if you go to the Wilmot shared resources page, here at the bottom, Wilmot Biobank and Biospecimen Research Center. If you click on that, it's our own biobank database. So like I said, much more details included on there, links to useful information, and a lot of links to get this process started, to request samples, to get onto our database. And if that doesn't work for you, please feel free to reach out to me, email me anytime. That's what I'm here for. So, most basically what we do, right? What services do we provide? Well, really the foundation of our resource is our prospective sample collection, right? And we call it our prospective collection because these are tissues, blood, and data that are collected prospectively for future use research, right? So each of the samples that we procure, we annotate with clinical data. And we can obviously release that data to you in a de-identified fashion to annotate your own studies. Beyond that, we really do our best to aggregate as many clinical tests and clinical reports as we can. So later I'll show our database. If what you're looking for isn't on there, we can absolutely do advanced data searches and pull together and de-identify a list of more disease-specific information, hormone receptor status in breast cancer, PSA, Gleason score in prostate, things like that. So beyond our prospective collection that, you all could start using tomorrow, we're also more than happy to support targeted collections, right? And really what we are able to do and what we're happy to do is any piece or any part of clinical research. So whether that's, a targeted collection of, a specific type of tumor you're interested in, specialized processing specific to your project, or even just cold storage or any combination thereof, we're happy to support. And, all the way from, the level of patient identification and consent down to analysis, especially with some of the spatial technologies we're looking to bring in that I'll talk about later. And, beyond these services, in a lot of what we do already is really support for this. Reach out to me as early as possible. That it's always best, so I can understand, and hopefully help, the design of your human research project, develop methods, even down to the level of, support letters for your grants, right? Especially, and this is where we're seeing a lot of interest, is in regulatory aspects. So, putting in a study protocol for your research project and working with the RSRB. That's a big part of what we do. To that end, I don't want to talk a lot about this or get caught up in the regulatory end, but I wanted to mention our study protocol because it's really the backbone of what we do. This is the first page of our consent that's tied to that. And it really, lays out in simple terms what our study is, what we collect, and what we're going for here. So the patients we look for are at Wilmot for a biopsy, a surgery, or a resection procedure of a potentially cancerous, or otherwise diseased organ. So that's a way to kind of extend a bit beyond the realm of cancer, both to bring in normal tissues and I think to an extent some other disease states that may be even loosely tied to cancer. We collect blood, so we also wanted the ability to collect specimens from patients who aren't undergoing surgery. So, for example, cancer patients who are here for a chemo treatment, and we want pre and post blood collections, right? And beyond that, also screening procedures, right? So this is another way to get, normal or even benign tissues, through colonoscopy or whatnot. But also, possibly get some data points early on in disease progression at first diagnosis that we compare with later resections or even metastatic recurrences, right? We're still building this up a lot, and I'll mention it a couple more times, but we really want to expand our longitudinal collection capabilities. So the purpose of this study, that we tell subjects and what we do with these tissues is we bank them for future use research, and to that end, undefined future use research. They'll be stored with clinical information to annotate them, and it's for an indefinite period, right? So that's how we, begin building up our prospective collection. Yeah.
So you have brought in this plasma of serum, and also do you collect urine?
Yes. So yeah, I didn't mention it. Urine is on here, and we're also, we're constantly amending this protocol, and we're beginning stool collections as well. So urine and stool are both on there.
And the blood, you collect the serum or plasma?
Yes.
Both?
Yeah. So right. So if there's enough whole blood, so there's a lot to this, right? I think when you're talking about the actual processing, right? If there's enough whole blood, we can isolate serum from non-heparinized tubes, and then plasma and mononuclear cells from a heparinized tube, right? My preference is to, whenever, we have limited amount of whole blood, is to do a plasma and mononuclear cell isolation, right? But yes, we have at this point several serum samples as well. Yeah.
So all the treatment, even the new treatment in our system, you will collect the product before treatment and the after treatment and follow.
Yeah. We try to. We're building this up, right? So right now we're doing our best, and with the resources we have to collect what we can. But absolutely, if there's a specific study or time point or something you're interested in, we can obviously start building in those collections, right? Maybe it is a pre and post therapy, right? Or maybe targeting more recurrent tumors or re-resections, right? We can absolutely build that in if we don't collect it, but in general, the scope, right? All the different primary origins that are present here, even if we just focus on solid tumors, there's a lot to collect, right? But absolutely, we want to collect what you're interested in. So if you come to us with something, if we don't have it, we can add it in and begin those collections. Yeah. So what I wanted to highlight here, in thinking about the regulatory side of things, right? I'm sure some of you have put in study protocols to get research, consent patients get research tissue and analyze it for a specific study. our consent isn't a traditional study protocol, right? It's for open-end future use research. It's a biorepository study protocol, so it's a bit different. The other way that you may have used tissue for research before is through working with clinical collaborators, right? So in the clinical setting, if there's excess tissue, and you can get that from a clinician in an unidentified state, right? So no identifiers with it. You can use that for future use research, right? So why did we go through all this work to put in this, biorepository consent? And the reason for that is because of, the state of the NIH genomic data sharing policy, right? So it's now determined that when you do genomics analysis on these human specimens, you're really creating identifiers, and the potential to re-identify those patients, those subjects, comes up, right? So in the setting where, say, you get a piece of tissue from a colleague that comes with no identifiers, but you want to do RNA sequencing on it, you're creating identifiers, and there needs to be a way to consent that patient, or if they were consented, to reconsent to do genomics research. We're taking that out of the equation by consenting all of these patients that we collect right off the bat. The potential future use research that we highlight includes genomics. So these patients have already consented for bulk RNA-seq, single cell-seq, all of those genomic analyses to be done on their tissue. There's no need to reconsent them whatsoever. That said, when you go to use our tissues, the RSRB does want, whether you're using identifiers or not, does want each of our users to put in a study protocol. But again, we're trying to lower that threshold and make it as easy as possible. So again, on our website, we have a study protocol template for Biobank users that has much of the information you need already entered into that protocol. If you're doing genomics research, you'll add in a bit about the data handling and put in some generic text that we highlight in there. Really, we want to make it as easy as possible. In the study protocol, just put what you're interested in studying, the assays you're using, and everything else is pretty much baked in. And like I said, if there's no identifiers, it's going to be an expedited review and determined to be not human subject research. So even though you have to put this in, it shouldn't be a scary thing, and we're more than happy to help you through the process. So, you have your study protocol in place. You're ready to receive some of our prospectively collected samples. What is that collection, right? How do we get it? So I'm sure a lot of you have heard, at least at some point, about Indivimed, our industry partner, or the University of Rochester Cancer Library. That's what this is, right? So Indivimed is now part of Crown Bioscience, and we talk about their group as the Wilmot Cancer Library. So this was, really an advantageous partnership, because even before we developed this infrastructure in our in-house biobank, this group has been collecting samples, and they really, bolstered our inventory before we even, began to advertise and disseminate these tissues. And the relationship is pretty good, and the content of our current bank is in reasonable shape. They're interested in collecting a pretty comprehensive list of cancer types. It's primarily solid tumors, but there are some limitations to what they'll collect. Again, it's not every type of solid tumor, and also the resection volume. The tumor has to be greater than 2 centimeters. There has to be enough tissue for us to share between the two groups, both academic and our industry partners. The other, bit of a limitation, it's great that for the cases they collect, they'll handle the patient identification, consent, handling the tissue. But the processing they do is, I think basic is a bit harsh of a term, but they'll do more routine processing. So formalin fixed paraffin embedded blocks, non-viably flash frozen tissue, and then, your blood derivatives. But like I said, I'm by no means complaining. We have a really great inventory already built up. These are the different samples that we already have banked from our collaboration with them. Where we come in are a couple of different areas. So what we want to do, and we're just getting this started now, is rather than, you know, for every tumor that comes in doing your standard routine FFPE flash frozen tissue, we want to do more advanced processing and really tailor that to what research is being done for that type of cancer, right? So you know, if there's excess solid tissue after we get an FFPE block, what do we want to do with that? And what suits our research community best? You know, we could do viable single cell suspensions of tumor and normal tissue. We could pair that with tumor interstitial fluid, which I think is becoming quite interesting. Or we could do other preparations, OCT blocks, xenograft processing, things like that. I highlighted this. What I'm really interested in, we're working on a collaboration with an industry partner that's in vitrification. So I think moving towards, when you're talking whole tissues, of vitrification preservation versus just flash freezing could be super helpful, right? You're freezing that whole tissue in time, and upon thaw, then you can decide what you want to do with it. You know, single cell dissociation, or, you know, fix it and put it in a block. You know, it really leaves you open-ended. In current approaches to preserve whole tissue like that with a slow freeze with standard cryopreservatives isn't that great, especially for tumor. You're going to lose a lot of viability. They've shown some really good results, and I'm hoping this collaboration keeps momentum moving forward. And if I have any colleagues too in neurology, I'd really be interested in talking to you. They're specifically interested in brain tissue, and at least in mice, they've shown that their techniques can maintain electrophysiology upon thaw too, which I think is really big and really interesting. So beyond, you know, what we're adding, I think, to our perspective collection, obviously, as I mentioned before, we're also happy to do targeted collections. So low yield resections or rare tumors, something that Crown or in the Wilmot Cancer Library isn't, you know, interested or will be collecting on a day-to-day basis.
The xenograft preparation
Yeah.
is the patient derived.
Yes.
So, so if we are interested in certain tumor and
Yeah.
how we need to have first protocol, then make a meeting, discuss with your group, and then see whether we can get those.
Yeah, I think that's really best too, you know, and as I mentioned, tailoring what type of processing is done based on the tumor type and the research being done in that community here. I think that's really important, especially for xenografts, right? In that scenario, I think it would be best if, you know, maybe not the whole department, but those prostate researchers who are interested in doing xenograft get together and we come up with a protocol, you know, for those collections. Because, you know, there are several ways to do xenografting, and what would be best for us, and I think the community in general, is if we approach a discussion and develop a standardized protocol for everybody to use. And maybe that's different from prostate xenograft to breast xenograft or whatnot. But I think starting that discussion with the greater, you know, research group is really the best way to go about it. Yeah.
Some kind of format of spaces, tumor is kind of later stage.
Mm-hmm.
So we have an autopsy and then kind of procedure to kind of get the very late stage.
Yeah, we don't right now, but that's a good point. And I think, you know, leveraging autopsy as well to get more normal tissues is a, you know, a good reason to look into that. We don't at the moment, but, you know, I think when we get a little more on our feet and get our day-to-day up and running, I'd like to look into that, and especially rapid autopsies too. That's really the best way, I think, to procure some of those tissues through autopsy. Okay, so that's our perspective collection, right? And, you know, what's in that? Well, I'm not going to go through it in detail, but we spent a lot of time trying to build a user interface, a database that, you know, any of our investigators can log into and really look at the current content of our collection, and, you know, maybe get some ideas of what they could study or what they'd be able to study with what we have. And absolutely, you can just reach out to me and ask, hey, you know, what do you have for frozen breast cancer tissues? I'm fine with that, but I really encourage you to take a look at our database. The link to it's on our website, and to get access, again, there's a link that you click on, sends an automated email to me, I get you approved, and then you log in with your URID credentials. So it's pretty easy to get there. And and like I said, the how we present the content, you know, I think it's ever evolving. We always want to improve upon it, but it's really, we try to make it as informative as possible, but as simple as possible at the same time, because there's a lot going on here. So when you look, I think the main thing to note, there's 3 tabs: an overview of our collection, some case, subject, disease information, and then sample information. And as you go from tab to tab, different filters will pop up on the left, and you can kind of dial in on what you're interested in studying or what collections you're interested in looking at, right? So here, you see, as you would expect, kidney, lung, colon, a large part of our collection is made up of, those primaries. But like I said, we have a pretty diverse collection. And on the right here, what I'm really trying to highlight are paired specimens, right? So in thinking about this, your research project, whenever possible, you want to control. So do you want tumor tissue that has paired adjacent normal tissue from the same subject, or a subject that had a metastatic recurrence and or synchronous metastases, and we collected metastatic tissue as well, or even just paired blood, right? As you gate down, these numbers will change, and you can really filter and begin to look into what tissue pairings you want from that same subject, right? So here, for example, we're looking at prostate tissues, right? So here's our prostate collection, 253 cases, 799 samples, and here's the spread. So again, you would be looking in this region here if you're looking for paired tumor and normal adjacent. Next, looking at the cases tab, right? This is information that's relevant to really all tumors, solid tumors at least, right? As I mentioned, we can do advanced data polls and get you more disease-specific information, but this is what the NIH deems as, minimum reportable information you should be looking at regardless of what type of cancer you're studying. It goes from, subject information to treatment or exposure information, and then, classification of these tumors, right? So here's your filters. What filtered down on overweight patients with a adenocarcinoma of the prostate. And then finally on our samples tab, the same idea, but here we're looking at sample information, right? So you have a carryover of the origin and classification, but then information on the collection, right? When was it done? What tissue type are we looking at? Blood, urine, tumor, metastasis, how it was collected, which is more important as we collect more and more biopsies. It's not just going to be a standard, resection procedure. And the other thing to note here is this tissue localization. And why is this important, right? Well, say you're looking at a primary pancreatic tumor, but you want to study its metastases. The primary origin is going to be pancreas. But if you're studying a MET, the localization is going to be something else, liver or lung, right? That you need to know that, right? Yeah, you're looking at pancreatic cancer cells, but the host tissue does matter and should be considered in these studies. So that's why, that's an important piece of information I think that's often overlooked in, defining your sample cohort. Over here to the right, I won't get much into it, but this SPREC chain, there's info about this on our website. It's really a chain that defines the quality control measurements that we make. So everything from cold ischemia time, warm ischemia time, centrifuge speeds and times, that's all recorded in the SPREC chain. And information on how to read that is on our website. Really comes in handy if you're having issues, or the quality of your sample you think is off, or for reporting in publications. And on the right, we're, beginning to annotate this more and more, but really from our FFPs for every tissue we collect, we want to have an idea of, okay, in this tumor, here's the neoplastic content, here's how necrotic it is. So when you get a tissue sample, you're, not just getting a 90% piece of dead tissue that is, generally worthless. So requesting these samples, the process, I've tried to make it as simple and easy as possible, both for our reviewers and for our users. So, after you reach out to us, we have a discussion about, RSRB, get that study protocol and get it approved. The request process from there is pretty simple. We made it through REDCap, and there's a link to initiate this on our website. It's a survey-based thing that asks a few questions about the study you're doing. You know, are you approved by the RSRB and what samples are you interested in, right? That's automatically routed to me and sent to our review committee, which contains representation from, you know, each of our research programs, clinical trials, and leadership, right? They're not here to stand in your way. They want to see your study go through. So we just need this information and want to make sure that something big isn't being overlooked, right? But this review process is quick. So I already clicked off it, and you know, upon their approval, you know, we'll work together and get those samples to you. So that was the first part of what I wanted to talk about. I guess, you know, we can take a breath. Any, you know, questions on that, on the more day-to-day, you know, specifics of our resource and what we do? Yes. Hi. Yeah, that's a really great question that I don't have information. Well, I don't know right now. We have information on that, right? And obviously these patients can ask to be removed at any time, and we have had that happen. I think it's generally good. I think it'll change as I mentioned, we want more longitudinal collections. So if we can start a front door consent policy, you know, like they have at Roswell. So any new patient, hey, if we end up taking your blood or doing a resection, can we use that tissue for research? I think in that setting where, you know, everyone is a little more, I don't want to say calm, but, you know, thoughtful of the whole situation, I think our accrual rate could be improved. But yeah, I'll have to get those numbers. Yeah.
Logistically, how does sample collection and storage work? Is it, you know, the surgeon that's on any given
Yeah.
case that calls you, or is
Yeah.
it, how do you get notified?
That's a great question. So there's a couple different ways. So the easier one to talk about is a targeted collection, right? And what I've really come to love is when a clinician comes to me wanting to do research because they know what clinic their patient is in. They often offer to consent that patient, right? That makes it much easier. If not, you know, we're more than happy to figure out that workflow. For a prospective collection, you know, our patients are consented, you know, prior to the resection procedure. And at least with our Indivimed or our Wilmot Cancer Library partners, they'll use Surgery Snapboard to identify, okay, here's an interesting case coming through. We need to intervene, try to consent this patient beforehand. And then the day of the surgery, if that patient was consented, their staff members will, you know, monitor the surgery schedule, you know, head down a bit early into the OR, and then take the tissue back to surgical pathology. After, you know, the pathologists take what they need for clinical diagnostics, then they set aside what's allowable for research, and we go from there. Them being in the OR really expedites the process and saves some of that ischemia time, right? Rather than relying on, you know, the clinical routine of bringing those tissues up. Yeah. Yeah. Yeah, so we have a number of benign tissues on our database. As with everything else, if there's something lacking or something you want to collect that we haven't, bring it up to us. We're more than happy to add that to our prospective collection and start building those in. And yeah, we have a number of benign tissues, but you know, as I said, we're allowed to consent patients that are here for, you know, some level of disease. So we can also consent, you know, perhaps epileptic patients who are getting a brain resection for a normal control, right? We have those capabilities. I don't think we've gotten to the point where we're actively building that part of our bank, but we absolutely want to, and we want to get it started sooner rather than later if there's interest, and we're missing something that could be useful.
So for the whole database, we are interested to know, say, okay, the patient after receive some treatment, they are responsive or
Yes.
non-responsive group.
Yes.
From our bank, it already have enough information. We can handle search and say, okay, you already collect those samples, but because of the treatment after 2, 3, or even 4, 5 years later, you know, those patients may be less responsive.
Yeah. Yep. We can look into that, right? So that would be a very advanced data pull, right? To look back in those records. The best information we can give you is what's been done clinically, right? So it's either a clinical note of, okay, this patient's recurring, this patient's progressing. You know, we could get a, you know, any MRI done or a CT or something like that. That's where we would absolutely need a study protocol, because then you're getting into the realm of identifiability. But if that patient's consented to our protocol, we can look into their record and follow it up over time. I mean, at the very least to track survival, but, you know, like I said, we can, you know, parse it out and look into details of when was that recurrence? What did the doctor say about that recurrence? Things like that. Yeah. Okay. All right, so that's great. So this is, you know, a bit of a shifting gear, but I wanted to talk a bit about human tissue research in general, right? These are precious samples, and we want to use them the right way and get the most out of them. And so with respect to that, you know, how I'm going to kind of frame this last bit is, you know, thinking about what can be done with this, but really focusing in on the second bullet, right? So there's nothing wrong with using these tissues for ancillary data, right? Whether you want to generate a hypothesis, to you know, begin formulating a study, or even, you know, you found a strong mechanism and you want to show that it's relevant in human disease. Absolutely, we want to support that. But what I think isn't, you know, utilized or approaches much is building a comprehensive study, right? Maybe that's an entire manuscript, right? A holistic study, or even an aim of a grant, right? There are ways to do that just with human tissue. Now, yes, you want to look at mechanism, you want to pair this ideally with some in vitro or in vivo preclinical work. But you do have the ability to build a study around human tissues. And, you know, just generally thinking, you know, two approaches to that pop to mind, right? One, you can have a really sophisticated sample set, right? So maybe it's a rare disease, or, you know, really unique and informative collection time points around treatment and recurrence. You can approach it that way, but you can also use more standard samples and use your analytical technique and really mine data to build a complex story and to do complex analyses. And I kind of view those as, you know, both sides of the coin. What I'm, you know, the example I'm going to give is the latter, right? So a standard sample, performing complex analyses and really building a manuscript out of that. And one of the reasons I want to highlight that are some of the initiatives at the BSR. So these advanced technologies we're bringing in are more than capable of getting this level and this quality of information extracted from these tissues. But also, you know, we're actively increasing the value and utility of our collection. So even though, you know, this study could be just, you know, your standard samples, your FFP blocks, I think we are beginning to get more interesting collection time points, you know, early in diagnostics, resection, late stage, and other processing derivatives that could even further boost the info you pull from these complex analytical techniques. So what I'm going to talk about today is a study done with, this manuscript was published using FFP blocks. So that these were samples from our biobank, but long before the infrastructure was in place. This was this, you know, and it's easy for me to talk about because it's a study I was a part of, right? But, you know, at that time, we didn't have any paired specimens. We really did the best we could with these FFP blocks to look at pancreatic cancer, right? And even though radiotherapy, you know, doesn't cause this robust response and increase survival in patients, we thought it did have an effect on the tumor and could really promote an immune response against that tumor. And that's what we wanted to show. We wanted to highlight that, hey, something's happening in these tumors. We just need to build upon that, possibly with a secondary treatment. I don't want to get too wrapped up in our findings. I'm more so using this example to show some of these analytical approaches and thinking about spatial omics and how to build a story just by looking at that in situ tumor, right? Yeah, so as I mentioned, just FFP slides, right? What we use these for were multiplex IHC, and this was, it's not outdated by any means, but, you know, more, you know, of your basic multiplex IHC. So six target panels, 3 of them, one looking at, you know, your tumor cells and your non-immune stroma, and then 2 immune panels looking at myeloid and lymphoid cells. We paired this with sequential slides that looked at the tumor stroma with trichrome stain, and then FFPE scrolls for genomics analyses, RNA sequencing, TCR sequencing. So, this is what I think people, think about instantly when you talk multiplex IHC. It's a beautiful picture, it looks great in a manuscript. It's great for getting funding, but, it's purely, subjective analysis, the quantitative data you're getting from here isn't that valuable. And that is in fact far from the case, right? But when you look at it like this and get lost in all the colors and all the different morphologies, right? I think it's easy to lose the objectivity underneath it all. And you have to really peel back these layers to see that. So that's what I'm going to do here in kind of walking through how this study was built, right? So a lot of these spatial analysis softwares are the same. In a lot of, these analyses I'm talking about are general across them, right? The way of thinking is I think what I'm trying to get across here. So taking away all those stains except one, right? A pancytokeratin, this is marking tumor cells. This is a pancreatic tumor cell nest, I'll call it. So based off of this, fluorescence, the artificial intelligence in these analysis packages is pretty robust. And this is easy for that software to do, to define, hey, here's the cell, to mark its nucleus, to mark its space within that tumor. And off of those objective definitions, you can perform your measurements, right? So in this case, tumor cell density, how many of these cells are present across that entire slide, right? And also you can look a bit at the characteristics of that cell. In this case, we looked at nuclear roundness, right? All measurements you can make with just one stain using this, robust software, right? And interestingly, here we saw that in fact, radiation did cause a decrease in tumor cells, suggesting that there is a stimulus, right? There is a cytotoxic effect of this therapy, even though we don't see a survival benefit. Then we start building up. So on the same slide, right, we added more markers. Here's a checkpoint ligand and essentially antigen presentation, right? Histocompatibility, MHC1, right? So here on the left, you're looking at the pancytokeratin. Here are your tumor cells. Here's this, and these are called segmentations, right? We're segmenting this cell population. This is checkpoint positive tumor cells. And then here we see class 1 positive tumor cells, right? Each of these together make up a greater amount of this tumor content. And now we can look at not just tumor cells, but checkpoint positive and measure them, and class 1 positive. And really, with respect to the data, what this told us, there are fewer tumor cells. Radiation is likely killing some, but those that are left are more immune suppressive. They're likely causing immunosuppression through presentation of the checkpoint ligand, right? Moving even a step further, so this analysis that we did on that slide looking at tumor cells and non-immune stroma, bringing in a second slide and registering it, aligning it with the first, we can bring in a whole nother series of markers, right? And this slide contained lymphoid markers or CD8 T cells, right? Your anti-tumor immune cells. These are the effectors most of the time, right? And what we did here was a similar approach. It's not shown, but we segmented these CD8s similarly to these, suppressive tumor cells and looked at their proximity. And we could gauge, okay, which of these CD8s are close to a suppressive tumor cell and might not be working properly, and what are farther away and, might be activated and might still be in an anti-tumor state. Building on this even more, bringing in a third slide with myeloid suppressors, right? We can do the same thing, but looking at more suppressive populations. So here we have our myeloid suppressors from one slide, lymphoid suppressors, T regulatory cells from another, and the tumor suppressors I mentioned. And we can look at CD8 T cell proximity to each of these suppressive populations, right? And what really stood out is there's not much difference, radiation or not, but looking at myeloid suppressors, they're in much closer proximity. They have a lesser average distance to CD8s and are likely the cells, if any, that are causing this immunosuppression and preventing that radiation damage from manifesting into an anti-tumor immune response. And then the last part of this, another sequential slide. This is not an IHC stain. This is a stain for the desmoplastic stroma of pancreatic cancer, right? And it's somewhat hard to see here, but there's a blue line outlining some of this dark blue region, right? What this is a training of the analysis software. So you give this artificial intelligence algorithm some examples of, okay, there's no collagen in this region. There's, light collagen here and dense in this region, right? You train it, you apply it to the entire slide. And what it can do is annotate and create these regions of dense collagen. They can be applied, as I mentioned, to the neighboring slide, the register slide. And then, looking at CD8 T cells and the segmented CD8 T cells in the bottom, you can see how many are adjacent to these regions of dense collagen or within them, right? And again, you apply to the whole slide and you can see here within that greater piece of tissue on that slide, you see in blue regions of dense collagen annotated, red T cells within, and green T cells that aren't within those regions. Why is this important? Because many thought, and still I think generally believed, that excess collagen can sequester and prevent these T cells from getting to the tumor and wiping it out with an immune response. In fact, what we show here is the opposite. After radiation, there's much more collagen in these tumors, but the number of CD8 T cells in that dense collagen is far lower. So this really suggests, and it goes against the argument that dense collagen prevents an immune response. This is actually showing that, in fact, most of these cells aren't in dense collagen and not being restricted by it. So again, this isn't about the paper, but it's about kind of thinking about this analytical approach, taking all of that information and even more that we extracted from these FFP blocks, right? The in situ human tumor. We put all of those findings together and really looked at the main drivers of these tumors and grouped them based on that using a PCA analysis. And what we really found was, indeed, there was a subset of human tumors that seemed to respond well to radiotherapy, that an immune response was initiated, right? The majority no, and that's what you would expect. But some did show an immunogenic cell death and the potential for an immune response. And possibly these cases would benefit from a combination with a checkpoint inhibitor or some other immune stimulatory therapy, right? What I didn't show on the previous slide were those last two sections. Remember, I mentioned we took FFP scrolls for RNA sequencing and TCR sequencing. Why I didn't show them is because all we had at the time were blocks. And even with our current specimen collection, there are far better things we can do and better data we can collect from these cases, right? Rather than doing sequencing on these blocks. At the very least, we have paired frozen tumor that'll improve the sequencing results, right? And paired blood. And when you're looking at, the clonal T cell response, with our blood, you could compare systemic immunity versus intratumoral immunity, right? It's a much more interesting assessment than just, bulk tumor T cell clonotypes, right? Beyond that, and this is where we're getting into these processes and procedures in the biobank, are viably dissociated tumor cells, right? So, simply for TCR sake, again, improve data. You can sort out CD4, CD8 T cells and do your sequencing on those. But on the solid tumor itself, right, like you have a couple options. One, you can sort those cells and do bulk RNA-seq on a specific population. Or you could do single cell seq, right? You have so many more options when you have more high value tissue processing done, or samples that are more amenable to several forms of downstream research, right? And then beyond this, and again, as I mentioned, to reinforce it, we're really trying to build our longitudinal collections, right? And that would be great for this study because maybe you collect blood over time and look at the expansion and contraction of the anti-tumor T cell clonotypes present, right? Maybe you collect tumor tissue later on, or there's metastatic disease and you get a tissue collection, and you can repeat some of these RNA sequencing or TCR-seq on that. And really, what this leads up to is being able to map and analyze the tumor microenvironment as it evolves over time, how it responds to treatment, how it begins to resolve, how it recurs. these both, paired sampling and longitudinal collection can really, I think, build to some interesting and really comprehensive studies just using human tissues. And then lastly, how this is all tied together. When this study was performed, it was a collaboration with Fred Hutch. At this point, we are now bringing spatial omics into our facility, and we can do it here. And why we really want to focus on this is because I've showed you all the information you can pull from one section of FFPE tissue, right? These blocks are easy to store. They don't require cold storage. You get 500 to 1,000 sections per block. There's so much information embedded in that. So it's really a great sample type to use. And when you're able to extract this level of information, it really, I think, pulls as much value as you can from these specimens. So with respect to spatial omics, where we're at, we just now have, it's in a crate still, unfortunately, but it's in our possession, a xenium in situ, right? So this is looking at the transcript at the single cell level, right? So it's not multiplex IHC looking at protein. It's really looking at transcript within a cell, what's present, and you can look at up to 300 to 500 different transcripts per run. It's a probe-based approach, and what's interesting about that, you can design your own probes to look at specific variants in your tumor of interest. These probes are ligated to the transcripts. There's a rolling circle amplification, and then part of that amplification is a unique sequence that binds fluorescent probes. So you go through iterative imaging cycles, adding fluorescent markers, imaging, washing, and repeating, right, to build these really robust, essentially 300 to 500 target images. There's not many publications using this technology, at least on the 10X website. But I did find one that I think really does a good example of just showing what this can do. Here's one cycle. Here's the fluorescence. Here's essentially transcripts in fluorescent colors across the breast tissue, I believe this is. The system uses, again, an AI-based segmentation program, right? This is coupled with an H&E stain done on the slide after the fact, but it essentially outlines where a cell is based on transcriptional pattern and, marks that off, right? And you can see all the little fluorescent dots in here showing the different transcripts. Pretty good resolution. And again, you're on average looking at just under 200 per cell. Again, there's 300 some on this panel, but on average, you're reading just under 200 per cell. And then this is where, again, similar high-level analyses can be done, right? And this is really where the value is in this sort of data collection. You take those transcripts in each cell and you define, okay, this is a perivascular-like cell. This is the transcriptional pattern, right? This is a macrophage M1 or M2 pattern, and that's super valuable. If there's any immunologists in here, you can't define a activated or suppressive macrophage by a few markers. You really need a good list to really define that phenotype and how suppressive it is. So then after you identify and annotate your different cell types, you can look at that image again, but not looking at transcripts, looking at cells that you annotated based on those transcript expressions. So, and then lastly, just to finish up, we don't have this technology yet, but we have the money for it. We have New York State money to support advanced technologies for specialized biospecimen processing and analysis. We're using this for multiplex IHC. So that study I showed you, rather than having to do sequential 6-plex stains, a lot of these platforms can support, up to 50 antibodies per panel. But we're taking our time with this because there are a couple different options out there. Some use a primary antibody and barcode conjugation for fluorescence, while others use standard primary and secondary that has some benefits that, for the sake of time, I won't go into right now. But, with those, you can build in DNA scope to look at the genome and really resolve some low-level proteins in those tissues as well. So to finish, again, we would very much like to thank Vivian Palladoro, who supported this lecture series. Without her and without Tracy organizing it, we couldn't have done that. And then here's a list of the upcoming talks for the shared resource schedule. So thank you. Yes. Yeah.
You talk quite often about, determining the quality
Mhm.
of the project.
Uh-huh.
Do you have some kind of way to make the decision? Who makes
Yeah.
the decision?
Yeah, so-
Whether you're actually saying yes or no.
Yes, so that's where the advisory committee that I showed before, our Biobank advisory committee reviews the sample request. What I would say to that is that's only really going to be analyzed and looked into if you're trying to request a rare specimen, right? So if there's, if you're looking for a colon cancer sample that's pretty ubiquitous, it's not an issue. But if, say we only have one tube, one aliquot of whatever tumor tissue or blood specimen left, then we'll put a little more thought into, is this how we want that used, knowing that it's the last one. I wouldn't worry so much about that. It's really to make sure that we have oversight for this process and that there aren't instances of that where we're losing or wasting, rare collections on research that isn't well developed.
The other question was, let's say you have two like studies that are
Mhm.
like similar things,
Yes.
and they're done independently.
Mhm.
Do you have any access to the data?
Mhm.
And see
Yeah.
any part of this to be like meta-analysis?
Yeah, we're working on that in a couple different areas, right? So first, it's doing our best and getting better at aggregating clinical testing results and pulling them in, especially, germline testing that's been done. We want to build that into our database as well. Beyond that, we've had discussions in building sort of a genomics library for these specimens, right? If we can secure funding for, a simple RNA-seq run or Sanger sequencing on these specimens, right? Building our own catalog of useful genetic information or genomics information to pair with these, we would like to do. I think we can. It's just taking us some time to build that and figure out how we're going to go about it. Yeah.
For the circulation tumor cell, this one will not need to kind of
Yeah.
walk and then see how the mutation coming to collect themselves.
Yeah. So again, circulating tumor cells, something we're very happy to collect. It's not part of our day-to-day right now, but if there's interest in that, we can absolutely make it happen. We're actually working with a study with Dr. Weiss, Anna Weiss, looking at circulating tumor DNA in breast cancer specimens, right? So similar idea, something, we collect the blood, right? And the DNA's in there, so we have it. Circulating tumor cells are a bit different. We would have to process them the day of, but again, we want to support the research, right? So you tell us what we should be doing on a day-to-day and how we should be building our collection. The more I hear that, the better I can do.
Yeah, when you talk about the FF,
Yeah.
RNA-seq and the PCR-seq.
Yeah.
You see, another advantage is you will be able to kind of get a viable with associated tumor. So those should be fresh collected samples.
Absolutely, yeah. So we don't have those in our stock right now. That's what I'm saying. So rather than, I think FFP blocks are invaluable, right? But beyond that, if we have extra tumor, what do we want to pair with that? I'm not of the opinion that just snap freezing that tissue is the best way. Yeah, it makes great bulk RNA-seq, but beyond that, what can you do? I'd much rather do a viable preservation of that whole tissue through the vitrification that I mentioned, or viably dissociate, right? And build a collection of single cell dissociations viably preserved for each cancer. We don't have those now, but that's, without any input, that's what we will be building in day to day, rather than generic snap freezing of these tissues. Yeah.
Maybe I missed it. So your paper published the paper
Mhm.
actually. So when you say you have a stain and 6 antibodies, do you have the one same line stain 6 colors,
Mhm.
and then you program this kind of to get all the far, right? Sure.
Yeah.
Just because you cut that same slide.
Yeah.
Visual stain different color.
Yeah.
The antibodies.
So yeah, a couple things there. One, when you're talking about several markers like this and co-registering and looking them at once, it's really pseudo coloring, right? There's not enough colors in the palette, or at least, visible to our eye that can annotate. You're really collecting all that data and then pseudo coloring as you go to look at different aspects of it. Our study, so at the time they had, these ultra high-plex IHC approaches were out. They were super expensive. So we stuck with the traditional way of, you're talking 4 to 6 markers per panel, but then using sequential slides, right? And I think the pathologist always told me like up to about, 20 microns in depth, you're really looking at the same section to an extent. So that's the approach we took, 6 antibodies per panel, sequential panels. And then, like I said, this analysis software is amazing. It registers it for you automatically. You just do a rough outline and it'll snap right to it. Yeah.
But you have the program.
So with this technology, we'll be bringing in these programs. And what I would like, again, whether we're using the Xenium for spatial transcriptomics, or we're using a different imager for multiplex, ultra high-plex multiplex IHC, these programs, in the end are really doing the same thing, right? And I don't know if this is possible yet, but I'd like to keep it to a minimum. Maybe we have 1 or 2 go-tos that we're well trained in, and we can teach you really the best way and get the most out of the data using those softwares, but to be determined. But yes, we absolutely will need a license for, omics analysis software. Yeah.
When do you think the Xenium will be up and running, and what pre-processing would have to be done? Like, how do the samples have to be stored in order to use it?
Yeah. So the great thing is, so one, if you email John today and start complaining, that'll help. We have it here, and I have more cold storage coming in. I need that thing set up. We have everything we need. I think we're waiting on a couple things, but it'll be very soon. The good thing is the input material is FFP blocks. The entire process, you're talking about, obviously you have to do antigen retrieval, deparaffinize antigen retrieval and whatnot, and then do a pre-amplification. So the actual run takes, I think 4 to 6 days. But with respect to the input material, one of the reasons we want to go this route is we already have well over 2,000-3,000 blocks in our storage, and there's so much information in there that we can pull this way soon. Yes, but we can talk anytime. It's time to start talking about it. All right. Please email me. Don't hesitate. Thank you.